My name is Jessica Lee. I'm an assistant professor in the Department of Statistics at UCLA, and also have affiliation in the Department of Human Genetics. And this work is related to my graduate work in the modENCODE consortium. So our work has been published in 2 papers. The first genome research paper is primarily about our work, and our work is also part of the consortium paper published in Nature in August. So first of all, let me introduce the question. So our question is very simple, but also sounds very crazy, because we're trying to study the life cycles of 2 species, model organisms, fly and worm. And here is their life cycles. So we know they are very different. So for example, the fly life cycle involves 2 genders, female and male, while worm has many hermaphrodites. So 99.5% are hermaphrodites, only 0.05% are male. And second, so worm has a special path called Dowers path, which is like hibernation. So when there's lack of food or it's too crowded, then they will enter this Dowers path. But fly doesn't have it. And third, there are far more cells in fly than in worm. So they are very different. But we are interested in whether we can find any relationship or correspondence or mapping between their life cycles. So this is our question. And the modENCODE consortium gave us this opportunity to study this question because they have collected a large number of RNA-Seq data at different time points in the fly and worm life cycles. So in fly, the data are generated by Sue Selnicker and Brenton Gravely in the consortium, and it covers 30 different time points in fly. Especially in the embryonic stages, the time point is very fine, so a point in every 2 hours. In worm, the data are generated by Dr. Bob Waterston's lab in Washington, and they also generated a large number of data sets for the embryonic stages, so in every 30 minutes. And in total, there are 38 stages with RNA-Seq data. So this gives us a great opportunity to study our question. And in addition, they also generated tissue and cell data for 10 fly tissues and more than 20 fly cell lines. For worm, they have 4 tissues and also about 8 cell types. So please note that in worm, the cells are dissected cells, which are not cell lines. So they are very different, and I'll show what we found. So our first question is, because we would like to compare the stages across the 2 species, then we should start from something simpler. So that is, within the species, can we find a clear mapping between its stages in terms of the gene expression, which we can learn from the RNA-Seq data. So here I just use 2 stages as an example, the embryonic 0 to 2 hour stage and the adult female plus 5-day stage in the fly life cycle. Okay, a quick answer would be, can we use correlation analysis? Because this is very commonly used in a lot of computational biology studies. So we start from that, and we compare the Pearson correlation between every 2 stages. So here we have, for every pairs of stages, we calculate a Pearson correlation, whose value ranges from 0 to 1. And we see, so there are a lot of stages with high Pearson correlation, and the 2 stages we are interested in have a moderate Pearson correlation. However, this does not give us a clear mapping between these stages. How about we try the Spearman's rank correlation, which is supposed to be more robust than the Pearson correlation. And what we got is a better pattern. So we can see that the stages which are close to each other have better correlation. So we can see that the correlation is higher near the diagonal. However, it still doesn't give us a clear mapping. Then what should we do? And why does those correlation analysis fail in our case, for our goal? So the reason is that all these correlation analysis use all the genes in fly, including those housekeeping genes, which are constantly highly expressed at all stages, and those will drive these correlation value very high. So that's why we see high correlation values between a lot of stages. And this is not desirable in our case because we want a clear mapping. So this tells us that we need to do some filtering on the genes. We should not use all the genes, but we should focus on some genes that capture the characteristics of every stage. So we should remove those housekeeping genes. And so we define a term called stage-associated genes. Here the term refers to the genes that are highly expressed at one stage, but it varies between different stages. So for example, these are two examples, each we picked up for one stage. So in this gene, we can see that it's highly expressed at embryo 0 to 2 hours, but it goes down at some other stages, which means that it's not a housekeeping gene, but it varies. And also this gene is highly expressed at this adult female plus 5 days, and it varies at other stages. So the criterion we use here is that we standardize the gene expression values into z-scores, and then require the z-score to be above a certain threshold. Here we use 1.5, so that the gene is relatively high expressed at the stage of our interest. And also, because there are some genes whose expression is low in every stage. So to prevent that, we also require the gene's absolute expression is above a threshold so that we are not using noise. And here we use cufflinks to calculate the FPKM and require the FPKM to be greater than 1. So please note that we have tried different values for our thresholds, and we get very consistent results. So we use this approach to select more genes. Here are 5 gene examples. And in the end, from the population of off-fly genes, we selected about 1,600 genes for the embryo 0 to 2 hours, about 500 genes for the adult female plus 5 days. Please note that here we call it stage-associated genes, not stage-specific gene. The reason is that we do not require every gene to be only specific to one stage, but a gene can be associated with multiple stages. So for example, in the case here, we can see these genes are associated with both the adult female stage and also some embryonic stages. So let me give a summary about how many genes we found for each stage. So in the fly stages, you can see that the number changes from stage to stage. In short words, we can see that the genes have a great number in embryonic stages and in the adult stages. The reason is that in embryonic stages, there are a lot of transcriptional activities going on, so therefore there are more genes that are highly expressed in those stages. And so that's why they have a greater number. Also, we divided these genes by the number of stages they are associated with. So we can see that most genes are associated with from 2 to 4 stages. This makes sense because we have a time course here. So the transcriptional activity is continuous rather than discrete. And some genes are associated with one stage and also its neighboring stages. And this is the C. elegans stages. We can see that, interestingly, there are 2 stages which have a larger number of genes only associated with the self. So this stage is one example, which looks very weird. And actually, we have contacted the lab who generated data, and they said that this sample has bad quality. So, which means that we have detected that. And this is another sample, L4 stage male. So this is the only male sample we have here. So that's why it's very special and different from others. And that's why it has more genes only associated with the self. And this is the fly tissues and cell lines. Again, we see there's one sample has a greater percentage of the genes only associated with itself, and this one is testes. This is the only testis tissue we have in our collection, and that makes sense. And this is the worm tissues and cells. Okay, so given those stage-associated genes, how should we proceed? So we're very interested in whether their stage-associated genes are 2 unrelated samples, or they are actually have some dependence. So therefore, we are interested in how large they overlap, which means how many common genes do these 2 sets of stage-associated genes share. And here's our approach. So again, we start with 2 stages, then using our criterion, for each stage, we selected their associated genes. And then we check their overlap, and then we design a statistical test, which is based on hypergeometric distribution, and then we calculate p-value. And then because we are doing multiple comparisons here, so we need to correct the p-value so that we don't have that multiple testing issue. And we use bumper running correction, the most conservative way here. And here's our result. So this is intra-species. So we're comparing the fly stages to fly stages. And we're very happy to see that we have a clear diagonal here. In addition, we also see a very strong mapping between the fly early embryo stage and the female adult stage. So we're interested to find out the reason for that. And then we resort to some database that we can find. So this is a database we find that annotates the fly genes. It divides the fly gene into 3 categories: zygotic, maternal zygotic, and maternal expressed genes. And then we check how our stage-associated genes are mapping to these 3 classes. We see that most of the maternal and maternal zygotic genes are enriched in our early embryonic stages and female adult stages. So this is the reason that we see that mapping. Moreover, we use this approach to compare the fly tissues and cell lines to themselves. So first of all, we see that the cell lines and ovaries form a block. They map to each other. However, the cell lines do not map to their corresponding originating tissues. So this is very interesting, showing that the cell lines are very similar to themselves, but not similar to their origins. Also, we see the head tissues are mapped to each other, regardless of their stage or their gender. And the digestive systems are mapped to each other. So later, we compare the fly stages to the tissue cell lines. The most interesting finding here is that the ovaries are mapped to early stages, embryonic stages, and female adults. Again, confirming that the maternal expressed genes are the key factors leading to the mapping between early embryonic stages and female adult stages. So we use the same approach to compare the stages within worm. And again, we see a very clear diagonal pattern. And also, we see a mapping between early embryonic stages and adults. As I said, the worms are hermaphrodites, so it also contains ovaries and eggs. So this is the same as the fly results. And moreover, we see that the 3 downward stages, because it's a special path, we put them to the right-hand side. We see the 3 downward stages are matched to the larvae stages. And this is actually consistent with the worm life cycle, where you can see that the worm enters downward from L1, after L1, and then exit into L4. Because during the downward stages, basically not much transcription happens. So therefore, the 3 downward stages are very similar to L1 and L2, which is what we found here, consistent. And we compare the worm tissues and cells to themselves. And first of all, we found that those cells extracted or dissected from the whole body worms, which we call oocells, are mapped to each other. And the cells which are from embryonic tissues map to each other. And there are also cells which are either from 4-cell embryos, which is the very early stage of embryo, and adult gonads. They map to each other, meaning that, showing that the very early embryo is very similar to the gonads in terms of transcriptome. And then we map the worm stages to worm tissues and cells. And what we found here is that the 4-cell embryos and the adult gonads, they are matching well with fly early embryonic stages and fly female adults. So we see that correspondence again. Okay, now we have seen enough good results for the interspecies comparison. How about our final goal, our ultimate goal, which is to compare the life cycles between fly and worm? This is our question 2. And we would like to have a clear mapping. And to do that, we use the orthologs to link the 2 species. So in our paper, we use the modENCODE orthologs in this paper. And because the orthologs have different kinds of pairs, we have 1-to-1, 1-to-many, many-to-1, and many-to-many. So to make our task simpler, we just decompose them as ortholog pairs, 1-to-1 pairs. And we use that as the link between the 2 species. So here we are trying to do the interspecies stage mapping. We would like to use the same approach. But here, because they are not, the two species do not have the same set of genes, but they have orthologs. So therefore, we use the orthologs to link them as the overlap. And similar approach is used to do the mapping. So one stage from fly, one stage from worm. And we select their stage associated genes independently. And then we counted how many ortholog pairs exist between them. And then we check the statistical significance. Then we correct the p-value for multiple testing. So very similar approach. And this gives us a very interesting result. Y-axis is the fly stages, x-axis is the worm stages. So we can see that, very interestingly, the worm life cycle is matched to the first half of fly life cycle. And then we see a very approximately parallel pattern again happening in the second half of the fly life cycle. So we just try to find this pattern by finding the line that goes through the largest overlap measure in each pattern. So we find these two parallel lines. So more specifically, for the first diagonal, we illustrate them as the one on the top. So we see that the worm life cycle is matched to the first half of fly life cycle. And then adding the second line, we see that it's matched again to the second half of fly life cycle. So this is very interesting, and we didn't expect that we can see such a clear result. And more importantly, we see that the fly life cycle is divided into two halves. And this division corresponds exactly to the time point where the fly undergoes metamorphosis. So during the metamorphosis, all the differentiated cells in the fly will be digested, and new cells will be differentiated from stem cells during the metamorphosis. So it seems like the fly is living two lives of the worm. And to better investigate why we have this kind of 2-to-1 fly-to-worm stage mapping patterns, we pick 3 stripes here. For every stripe, we investigated what genes are involved. So first, we look at the red stripe here. So in this stripe, we divided the genes into different categories. The genes only associated with the later fly stage and the earlier fly stage, and the genes in common, and which genes are different in fly, but they have the common ortholog in worm. So we have 4 categories. And we see that in this case, we have a pretty good overlap between the early stage associated and late stage associated fly genes. We check this with the within fly stage mapping. And exactly, we have a moderate mapping between these 2 fly stages. So we see that in this case, the mapping within fly is the key factor for this parallel pattern. How about the second stripe, the green stripe? We also did this analysis. Now we see that the brown part shows that there are greater number of fly genes that are not the same, but they are orthologs of the same worm gene. So we check the within fly stage mapping results again. We see that these 2 stages are matched with fly, but also there are some orthologs that are multiple copies in fly, but 1 copy in worm, also contribute to this 2 parallel pattern. The third stripe, the blue one, we see that now the overlap between the fly genes is small, but the number of fly genes that have common worm orthologs become greater. So therefore, by checking the within fly stage mapping results, we see that these 2 sets of fly stages, here and here, they do not overlap in fly. They do not map to each other in fly. So therefore, the main contribution here is actually these multiple fly to worm orthologs. So this tells us that because fly is a more complicated organism than worm is, so that's why the same gene in fly, after the speciation, it becomes duplicated, and they have different copies in fly, and every copy has a different function. So some copies function in the early fly stage, some function in a later fly stage. So moreover, I'll just go quickly about the other results we have here. So by comparing the worm tissue cells to fly stages, what we found is that the worm 4-cell embryos and adult gonads are mapped to fly early embryonic stages and female adult stages. And by comparing the worm stages to fly tissue cells, we see that there are fly adult gonads and fly cell lines mapping to worm early stages and worm adult. By comparing the tissues and cells between the two species, we have very interesting result. The first result is that the fly heads and fly CNS systems are matched to worm neurons. So this is very interesting. And moreover, there are fly cell lines and gonads matching to worm 4-cell embryos and gonads. This is what we expected. So here I'll just show one functional result. More results are in our paper. So in this result, we find the top enriched GO terms in the fly early embryos and fly female adults, and to see what they are. Interestingly, they are related to mitosis, DNA repair, and meiosis, and also DNA recombination. And we see they are very similar. And also, these GO terms are specifically enriched in fly early embryos and fly female adults. So this is why they are mapped to each other. And moreover, we try to use the same approach to do the stage mapping, but no longer relying on genes, but using the GO terms. So we are trying to use the overlapping GO terms to do the stage mapping. And here's what we get. So using the GO terms, we see a similar pattern in fly, but noisier. So those off-diagonal signal become stronger. In worm, similarly, a similar pattern, but noisier pattern. How about between fly and worm? So in this case, we get very little signal. The reason is that the two species have very different GO vocabulary. So if we use the GO term to link them, it's not very successful. Okay, so to summarize, so in this study, we proposed a new measure for comparing transcriptomes, and we named this as transcriptome overlap measure, short as TROM. And we will distribute our package very soon, and requests can be sent to me, so if you want to try it right now. So why this is useful? Because here I'm just repeating the result again. So the left-hand side is our measure, right-hand side is Pearson correlation. You can see a much clearer pattern can be obtained with our measure, and also C. elegans versus Pearson correlation, and also fly Spearman correlation, and C. elegans Spearman correlation. So you can see that without our measure, the clear mapping we found cannot be established. So conclusion, first of all, we found conservation in the development of flying worm, and we found two parallel collinear patterns. This is the first time finding. And we found strong orthology in maternal expressed genes, and this led to our mappings observed between embryonic stages, tissue cells, cell lines, adult stages, and gonad tissues and cells between the two species. And the cell lines are vastly different from corresponding tissues. Dissected cells in worm, however, they show great similarity with corresponding tissues and stages. And we provide a more effective approach than widely used correlation analysis for this goal. Future directions, here I'm just listing two. There are actually more things, a lot more things can be done with this data. So first of all, it will be interesting to conduct a functional study to better understand the underlying molecular biology mechanism that leads to our findings. However, a big obstacle is that the two species are so different. So they have very big discrepancy in their GO vocabulary. And the second thing is to consider splicing regulation in refining our mapping results, or possibly network study can be involved. So this is our paper if you need more details. And finally, I would like to thank my PhD advisors, Haiyan Huang and Peter Bickel, who are both statisticians at UC Berkeley, and my collaborator, Stephen Brenner, who is a biologist at UC Berkeley. And also I would like to thank Dat, who was a master's student who did some of the GO term analysis, and Mark Began for his comments on our paper. And I'd like to thank fly and worm modENCODE people, so Sue Selnaker, Roger Hoskins, who generated the fly data, and Robert Waterstone and Ladina Hillier, who generated the worm data. Thank you very much.
Okay, thanks. We don't have time for questions, and we'll move right into the next talk, which is Yuheng Liu, our next